Hello, everyone. Welcome to the final session of Day 6 of ACM India Summer School. We are pleased to have with us Professor Krishna Prasad, whose work bridges human cognitive learning and decision making. His ongoing research projects explore the behavioral and neural foundations of statistical learning, as well as perpetual and value based decisions, using a rich toolkit ranging from psychophysics, eye tracking, EEG, and fMRI to computational models like reinforcement learning and Bayesian approaches. They bring a deep, multidisciplinary lens to cognitive science. Currently, they are also applying machine learning to structural MRI data for the early detection of Alzheimer's disease. Please join me in welcoming Professor Krishna Prasad.
This is audible. Okay. So, can I just know the audience are already familiar with machine learning? Yes, deep learning. Yeah, and brain, neural network. Okay. So, I'll be talking a lot about the brain. Okay, and if you want to apply your machine learning techniques to brain data, then how can you use the domain knowledge to implement better algorithms, right? So we are not modifying the machine learning algorithms, but what we are doing is we are understanding the domain data and then trying to see how you can apply it in this domain, right? So, to very much begin with, okay. I'll just give you a brief overview for the benefit of students. I have a BSc, MSc in electronics and an MTech in artificial intelligence. Okay, this is many years ago, and then from there I moved on to do research in neuroscience. And the main motivation is, as an engineer, I would dream about building the robots. And this is the robot there that you can see. This was the humanoid robot with 48 degrees of freedom. So something like that uses very simple algorithms like imitation learning. So today, you see the latest deep learning algorithms use reinforcement learning, right? So where with multiple trials, a robot like this will learn to walk, run, jump, and do other things, right? This is a physical robot, by the way. This is not a simulation. And in those days, of course, we didn't have such an advanced reinforcement learning algorithms, but what we were using is a very simple brain-based algorithm called Hebbian learning, which is imitation, right? So you would have heard this neurons that fire together, wire together. That means if there is there are two neurons, both active at the same time, the weight between them is increased, right? This algorithm you are familiar with, right? So you are increasing the weight between two neurons if both are giving the same output, right? So the strength between them is increasing. This is how close you get to the analogy of the natural neural network and the artificial neural network, right? So when you say neural network, we are really referring to the artificial neural network and not the biological neural, right? So the whole idea here is we use a variety of techniques, right? And they all stem from basic understanding of psychology and vision, right? So one of the questions that I would always ask is, can computers see, right? You can have the best cameras there, but they cannot see. We can capture the images, right? So there is a difference between a sensation and perception. The sensor, the camera, is only able to receive, right? And then on top of that, you build models for object recognition, object localization, bounding box, and so on, right? So you are applying algorithms on top of the basic sensation. So the perception aspect is missing. So you cannot actually say that the computers can see, right? So obviously we would like to study the human brain because that is very effortless for a human to locate an object, to direct my hand, to adjust my grasp is a very effortless task as a human for us. But if you would like the same thing to be done by a robot, it becomes very difficult. So we would like to understand the human brain, and then the whole idea is how does the human brain function, right? So the brain functions not in a very well-defined way. What do I mean by that? You see, like it's like people trying to decipher the function of the brain, but they cannot get a complete picture of it, right? So it's like the blind person says that the like a pillar, right? Because see the animal in totality. So the same thing happens with the brain because it's a lump tissue, right? If you talk about your digestive system, if you talk about your cardiovascular system, there is a structure to function relationship, right? So the heart has so many valves, so many ways it pumps the blood, the lungs take in the oxygen, they mix with the blood. So the function and structure have a relationship, but that does not happen with the brain, right? And to study the brain, there are many, many methods, right? You can actually do invasive recordings of the brain by, implanting an electrode, right? This is what Neuralink is trying to do, okay? That is an invasive method, okay? So it's basically, if you want to control somebody's brain, maybe for therapeutic purpose or something like that, then you would do surgery and implant an electrode, okay? And if you don't want to go that invasive way, right, non-surgical procedure, you can have this sort of what do you say, the cap, which is recording from the surface of the brain, right? So from the scalp, you can basically record from the scalp using very simple devices like this. Just put it on the head of the person, or you can do more detailed scans of the brain using MRI, right? So all of these have different advantages and disadvantages. Before we look at one of those techniques, which is the electroencephalography, the recording from the sensors on top of the brain, we will look at a little bit about the brain, okay? Especially the visual system. So the visual system, basically we think the eye, which is your sensor, right, is acting, it includes a lens and it acts like a lens, right? And it falls on the retina from which the information is relayed to the brain. Now the magic starts once it reaches the brain, right? So when the signal reaches the brain, what is happening is there are what is known as a receptive field. Receptive field is basically where in the outside external world I am focusing a particular rods and cones of the retina or the part of the brain, the neurons that are responding to it. If it was not the case, there would be a lot of chaos, and we would like to avoid any chaos and any potential damage. So the way it works is two approaches. It's like a torch light, right? Or I would say it's like a laser light. So when you have a laser light, you are only focusing on one point. It's called on center and okay. So on center off surround means you will respond to a focus point, but surrounding you will ignore. What is the advantage of this? We are connecting dots. Okay. When you connect dots, your coordinate geometry says if you have 2 points, you can make a line. All right. So what the brain is doing is edge detection. Okay. How does it do? Orientations. All right. So when you have an edge, there's also an orientation to it. And this happens in the part of the brain known as the primary visual cortex. When I say any part of the brain as primary, what it means is it is directly receiving the input from the sensors. It's your first layer of a neural network, right? The first layer, you will call it as a primary. And here, what you observe, this is a Nobel Prize winning work of Hubel and Wiesel. What you observe is that the different cells of the primary visual cortex are oriented, are tuned to a different orientations. Okay. So each angle is covered. So if any of you are doing computer vision, you would know histogram of orientation gradients, okay? HOG features. That is based on this principle. So the brain is having different orientation tunings, and this is, if you see, each color is one orientation, all right? So, but in each of these colors, you see a large patch of the brain has the selectivity to different orientations. Okay. So horizontal, vertical, slanted, left and right. Why is there a lot of redundancy in the brain encoding? It is because if some the brain, then you actually don't lose all the information. Okay. So this is also the kind of architecture in neural networks when you do what is known as an ablation study. Ablation is a neuroscientific term. Ablation means removing something. Okay. So if you remove some neurons from the brain, then also the function remains intact. So that is why this is happening, right? Now, the important aspect is that the brain is a parallel processor. What do we mean by that? It's not doing a serial processing. So early day neural networks were called parallel distributed processing. That was the name that you would give to connectionist networks. The whole idea that we are getting from the brain is that you are doing parallel processing. Now, how is the parallel processing happening? So, majorly there are two different functions. Okay, one is the location of the object, the other is identity of the object. Right? You need to know both. It's not enough to know that there's a bottle to the right. No, sorry. Any one of the two information is not enough for me to go and reach and grab. I need to know both the information. So I need to identify it's a bottle. I also need to identify that it's on my right. Okay. So both the information are processed by different parts of the brain. Okay. So the ventral pathway or the pathway which is at the bottom of the brain is doing object identification. The dorsal pathway is doing the spatial localization. So it's a parallelly, the information starts from the primary visual cortex, and parallelly you are having dorsal and ventral streams. Now, that is good, but what information or how do neurons encode certain specific things? So there are two brain regions that are shown here. The PPA or the parahippocampal place area and the extra striate body area. So what happens is anything that is related to a place, okay, will be activating this region. Anything that is related to your body will activate this region. In fact, there's also a region in the brain called the face area. So whether it is a human face or a monkey face or maybe your pet's face, there is another region in the brain that specially encodes the faces. What does this tell us? That there is a modular architecture, right? So there are several modules in the brain. The brain is a hierarchically processing. It does parallel processing, but it also does have a lot of specialized modules there, right? Okay, I'll skip most of it because there's a lot that I would like to give a broad overview for you. So if you look at the structure, right, there is some broad, like there is a visual cortex, there is a motor cortex, there is an auditory cortex. So what you see is that the different senses are being processed by different regions, right? And you also see that there is a somatosensory cortex, which is like the input, right? The touch perception, and the motor cortex, which is like the output, which is the execution part, right? Movement part. There are 100 billion neurons in your brain. So if you want to really construct a neural network as big as the brain, then the supercomputers won't be enough. And each of them make about 10,000 connections each. And the total connections is greater than the telephone network of the entire world, okay? So if you just 100 billion into 10 to the power of 4, right, into 10,000, that's the number. Everybody would have seen this neuron structure. This is the basic single node that we use in artificial neural networks. There is an input, there is a accumulator, right? And then there's a threshold, and then there's an output. It's as simple as that. We are mimicking the biological neuron. And it's an electrical signal, or more precisely, it's an electrochemical signal. So, and the most important and interesting part of it is that it's a binary signal. 0, 1. So the shape of the neuron firing, what you see doesn't matter. It will always be the same. Whenever a neuron fires, it's 1. Whenever it doesn't fire, it's a 0. It's just binary, right? And now all your electronics, all your computer organization, architecture, everything comes in there, right? And the brain is just doing this. Zeros and ones. And that's what the artificial neural network that you design would also be doing. It just has a 0 and a 1. All right? Just the combination of all of that is what gives rise to a lot of wonderful outputs. We won't need a lot of terminology, but just show you this, how the connections are organized, right? Very, very complicated network. Many regions of the brain, hierarchically organized. Okay, we won't need to know the neuroanatomy part, but we will start to discover how we study the brain function, right? So we would be referring to what is known as an event-related potential. Brain signal is electrical in nature, right? Electrochemical, electrical, right? So, and what you can do is you can put electrodes on the scalp and just directly record. That's what we are doing. So, in response to a specific event, if there is an activity in the neurons that can be captured by simply having this electrical conducting wires, right? So what we do typically is you have what we call as a stimulus. Stimulus is anything that you perceive. It could be a sound, it could be a picture, it could be a movie, right? It could be a touch, anything that is happening. Or and then your brain processes it, and then you generate a response. Okay. If I ask you what is the color of this pointer, right? Then you would respond it saying black, and my question and the object was the stimulus. So the way we put these electrodes is basically very symmetric across the scalp. Okay. Now remember, recording from the scalp not the same as recording from the brain, but we are doing it because what is the reason? Non-invasive, all right? Frontal, central, parietal, occipital. Z is your zero line, okay? Even numbers, one hemisphere, odd numbers, another hemisphere. The number indicates how far are you away from the central line, all right? 3 is more closer than 5 or 7. So what happens when I ask you to look at this? You're saying that this is your ID card, right? That looks like you're doing it instantly, right? But that's not the case. It's not instant. First, you will look at it, then you identify it, then you will read it, then you will respond. There's too many processes that happen inside your head at a very, very fast rate. And that's what we are capturing here. And each of these processes is very nicely and beautifully marked with a peak or a valley. Okay. The upper peak we will call it positive, the valley we will call it as a negative. It doesn't matter because you know potential difference is always relative. Electricity is flowing means there is a potential difference, right? So we won't really bother about the nomenclature. You just use it as a convention. So all the different processes that the brain is doing, you can actually pinpoint them, identify them. In as simple as, okay, tell me what is this, right? A very simple visual perception will tell you how the brain is working. And if there's a problem, obviously you can target that. One simple and very find with these is every time you are surprised, they'll say, na, expect the unexpected, like your quiz questions. That time you get this, what is known as a P300 signal. Okay. This is a very robust and most commonly used signal for brain-computer interface because there is such a huge difference in the amplitude. Now, you can ask me, sir, you said it is only 0 and 1. How do you get different amplitude? It is, no, this is not, no, if it crosses threshold, it's 1. If it doesn't cross threshold, it is 0. This is the output of the neuron, right? So what happens is this is not single neuron, it's many neurons together. So it's a regional activity. So many ones together give you this amplitude, all right? So that's why the EEG signal, you will have the difference in the amplitude, okay? We don't have to bother a lot about it, except for the fact that the brain, especially the human brain, I mean, it's also applicable to other animals, is too big, okay? That is what, you say our intelligent behavior is coming from. And our skull is too small to fit the whole brain. So what happens is the brain is like, crumpled and fit forcefully into the brain, into the skull. All right? It's like a sheet of paper, a thick sheet of paper, folded and fit inside. So what will happen is that there is some deep parts, like you see there, and there are some surface parts. So when you are measuring from the scalp, remember that you will only get the surface part and not the deep parts, okay? Why it's important, I'll tell you. Again, one more main way one would think about these brain signals is in terms of the frequencies, okay? Delta band and theta band are like very slow frequency, okay? It's like a very sleep type of a stage. Alpha is when you're awake. Beta is when you're doing some tasks, like, you're attending the lecture, right? So if I put a EEG headband on each of you, okay, and then you are like not listening and going to sleep, then your delta band will be active. But if you're listening to my lecture, then your beta band will be active. So I can easily catch whoever is paying attention and who's not paying attention. And gamma is like a very high frequency. It's difficult to understand the functions of it because you're really talking about 30 hertz. 30 cycles per second is really fast. Now, when you talk about frequency bands, obviously the whole point is that if you do Fourier transformation, then we can use it to reconstruct the signals, right? And it's also used for filtering purposes, right? If you want to remove noise, you just remove high frequency components. It's a very basic signal processing. Now, what you do with this EEG is like there are these stimuli, right? Like I will show you pictures of bottles, pictures of chair, of fruits, of birds, of animals, okay? I'll call them as stimuli, okay? And then what we will do is just a brief amount of time, about 800 milliseconds after the stimulus is shown, and maybe about 100 or 200 milliseconds before the stimulus is shown, you take that segment of the EEG data, okay? And you can remove any artifacts. Now, what are the artifacts? What is not signal is noise, and this is very important for us, okay? And we need to remove that. One source is eye blink. Eye blink is a muscular movement, all right? Any movement of your jaw, all right? All these are much larger signal compared to the brain signal. See, brain is sitting inside your skull and you are measuring from outside. So technically, any movement in your face is more directly connected to the electrodes here, right? And also remember, the brain is inside what is known as cerebrospinal fluid, right? So that is like a cushion. It's like your mobile phone's flip cover, right? So that when it falls, the shock is absorbed, right? So we need to take out those artifacts, any muscular movements, eye blinks, and so on. And then what we do is we average the data. Okay. Now this is a very important point. When you average, the noise part of the signal will average to 0 because noise is random. Okay. And the signal will be not random, it will be systematically varying. So you will actually get the signal. Now this is a challenge for brain-computer interfaces. Why is it a challenge? Suppose I am having a wheelchair that I want to control with a brain signal. Okay. And then let's say you want to move forward. You want to give the command from your brain signals to move forward. So when you give that command, you are really saying it once. That is called a trial. But that is noisy. So you have to tell 10 times, go forward, go forward, go forward. Then the wheelchair should average over 10 trials and then it will go forward. Just see the difficulty now, all right? Maybe I'll request you, can you pass on the bottle to me, all right? Imagine me telling 10 times and then you responding, right? So when you design a neural network also, what do you say? Sir, I had very less data. We had only 400 images. I have only one trial. If you want to really think about, converting your thoughts into action, it only happens once. Your large databases like ImageNet is millions, right, of images. Having the big data is not at all possible when you're talking about brain data. You really don't have that, and you don't want as well, right? Because you want to convert instantaneously thoughts into actions, right? And a lot of generalization. This is another thing that the brain does very well, all right? And neural networks, the artificial neural networks don't do very well. Sir, I trained it on chest x-rays. It won't work on MRI images. Sir, I trained on MRI images. It will not work on face recognition, right? You may have designed an attendance system. There might be an app that you can design, but it will only do that. So the generalization ability is very less in the artificial neural networks. So I'll skip this. Yeah, just to give you an idea, I retained these slides just you see the type of signals you get. Because I think a lot of students do these projects on emotion identity and BCI and so on without really understanding the signals, right? So that's what I would like you to take away from this lecture today, right? So if the signal is noisy, then we follow the rule of garbage in, garbage out, all right? If your training data is bad, your output will be bad. That's why we need to really consider that. There are so many factors. Okay, this is drifts over time. Your signal is not stationary, right? Over the time, the electronics gets heated up, the person gets bored, the connections become loose. How do you correct for that? you would use, like, let's say a BCI task, you have some cross validation, right? So let's say you use the 8 trials for training and another 2 trials for testing, okay? 80-20 split. Now, what will happen is this 20% of the data is not like your 80% of the data. Your algorithms will work properly only when the data is distribution is comparable between the training set and the test set. If that's not the case, then how would you do it? Now, there is one more very interesting aspect, okay? I think I'll just skip. Yeah, we have a very good acronym here, commonly recorded artifactual potentials. Okay, so in order to do artifact correction, I would like to suggest a method called independent component analysis. Now, you might have heard the word principal component analysis, PCA. This is a cousin of that. This is a cousin of PCA. What is the difference between PCA and ICA? PCA is used for dimensionality reduction. So if your data is mostly distributed along a particular dimension, you would take that dimension as your axis of the data. So you don't have to worry about many features. You can only use those features which are mostly varying in order to build your classifier. Okay. Now, ICA, what it does, apart from doing these principal components, it will ensure these components obey the rules of your coordinate geometry. So in coordinate geometry, what happens? You have x-axis, y-axis, and z-axis in 3 dimensions, let's say. They are all perpendicular to one another. Why is that important? Because the relationship between perpendicular axis is not there. They are independent of one another. So that's why we call them independent components rather than principal components. In a PCA, it doesn't, that constraint is not there. It doesn't have to be independent. Now, the way you do it is obviously, let's say you've got many sensors on the scale. You can do an ICA, and anything that is a source of noise like eye blink, etc., can be identified and removed. Okay, and then you reconstruct the signal using only the cleaned components. All right. So it's like I have I think in chemistry you study this sedimentation, filtration, that kind of a thing, right? So you have impure substance, you use certain purification techniques, and then you separate out the pure component from the impure, right? So that's the idea between the signal and noise separation here. The logic here is corresponding to what we call as a cocktail party problem. Let's say all of you are talking simultaneously, right? And I will say one at a time. Why do I say one at a time? Because there are many sources and I don't have so many receivers here. So in this situation, what we will do is we will actually put many microphones. Okay. So if I have as many microphones as there are sources, then I will be able to, what do you say, separate out which source, which microphone. So what is closer to you and what is closer to her, will give me different amount of signals, but yours also will be heard there, right? So when you do the EEG based brain computer interfaces, you need to be aware that all the channels, all the sensors have the same information. One will have more information, another will have less information, depending upon how close it is to the source. So the ICM mixing and unmixing matrix does that aspect. Okay, nice. So that was an eye blink. This is muscle movement. These are, by the way, called topographic maps. It's like a interpolated smooth map. Between each dot is where you have a sensor from which you are recording from the brain. Okay. I just put in too many. Imagine your data is like this. If you clean, it may become a little bit nicer. Okay. Now I've already talked about this, right? There are different ways you can do surgery and implant something like this. You can just record from the scalp. You can also record from the muscles themselves directly. Okay. So if you want to use that signal, then depending upon whether you are recording from single neuron, bunch of neurons, or the entire brain, you have different resolutions. We will not go into the history of the EEG. The signals look something like this. This is what we can do with analysis. It has spectral means frequency. It has temporal time, and it has cognitive information available, the cognitive process that's happening. Now look at this. I talked to you about the P300. What is happening in this? You have horizontal and vertical highlight on a 6x6 grid. Okay. So each row will highlight once, each column will highlight once. You are focusing on one of the letters. 6x6 is 36 possibilities. English alphabet, 26 alphabets. So you are focusing on one of them. The one that you are focusing will be highlighted how many times? 2 times. Once horizontally, once vertically. Total number of highlights is how many? 12. 6 horizontal, 6 vertical. Out of the 12, 2 times your specific focus of attention is being highlighted. This is an oddball. Surprise. Find the odd one out. P300 signal. So in this fast flashes, at this fast flash rate, you are measuring the EEG, okay? And this spelling that you see in that graphic is generated from the EEG signal. So this is the most commonly used BCI paradigm called the P300 speller. It's easy for English because only 26 alphabets. If you want to do for Hindi or something else, good luck to you because it's 50 plus alphabets, so it's more harder. Now I have a similar display, a lot of flashing, right? All these Diwali lights happening. And you are supposed to focus on one of them. What do you think is happening there? Each alphabet is being flashed at a different, slightly different frequency. Very different. right? They are not all flashing at the same frequency. Each one has a small difference in frequency. Now, wherever you focus your attention, okay, that frequency will be reflected in your brain waves. Like resonance. All right? This is called steady state visual evoked potential. Okay? SSVET. This is the second major paradigm for brain computer interface. Another example. So in this picture, what's happening is that the person is blinking their eyes and we are measuring their EEG, okay? And it's easy to measure the blink because I said it's a very strong signal. And with that, you're opening and closing the robotic hand, right? Simple. It's more like a class project. This is complicated. This is called mental imagery. Now this monkey, of course, we are not doing EEG. We are actually recording from the neurons inside. And the monkey is imagining, and that is being translated into the robot's hand. So the monkey is not eating with its own hand, but it is thinking, and whenever it thinks, that gets translated into the robot moving. Now, this is interesting because potentially if somebody has a paralysis or is not able to use their hands, then for such kind of people, you can use this brain-computer interface using this simply thinking. So thinking we are calling in our technical terms as mental imagery. So this is the third major paradigm. Now, remember I mentioned that anything deep inside the brain, I will not be able to measure. If you look at mapping of our body on this brain region, hands, mouth and tongue, they are all properly represented. When it comes to feet, you see that it goes into the middle portion, right? You have two hemispheres and it's going into the middle portion. So it's deeper in the brain. So if you want to decode leg movement, it's going to be a lot more tough. But hand movement would be easier because it's on the surface. Anything that is deep inside will be tough. And also one very interesting fact is right side of your body is controlled by the left part of the brain and left part of the body is controlled by the right side of the brain. So if you look at left and right, your motor regions, that is in to be able to whether you want to go left or right. So it's a very, very far apart, very clearly distinguishable signal. So that is what we use with this motor imagery paradigms. So one simple thing where we were not doing motor imagery, but we were doing motor execution. That means you are actually opening and closing the hand, okay? And while people do this, you can get these landmarks from any computer vision library. We used MediaPipe to get those landmarks. And then you can identify if the fist is closed or open, okay? And now, at the same time, we are recording their EEG. And the hope is that we will be able to decode. See, left versus right, I said, is easy. But opening versus closing is the same hand. It's a little bit difficult. So the hope is that we'll get there to do that because that's very important. Okay, this is Munna by MBBS. If there were any NEET aspirants, you know what it means. The whole idea is about whether you think somebody is brain dead or not, okay? And if the brain is responding, Like, for example, this was a patient in coma, and this person was not responding to anything the doctors were saying. Now, what happens is we are instructing this person, but the person is not replying back, right? So we look at their brain activity. So she was asked to imagine opening the door, some movement related, right? Or playing badminton. In both the cases, there is movement, but the brain regions are very different, right? And in these regions correspond to the same thing that the healthy person's brain also. So although this patient was in coma, it actually doesn't mean that they are not responding. They are basically not brain dead. I'll not go into a lot of details. I'll just give one example. So this was a very early attempt, 2011 paper on reconstructing a movie that the people saw while their MRI scans were recorded. Now, this is very tough. You can see that it wasn't very great, but this was pre-deep learning era. Now, this is the work of Prajwal, the PhD student, and with me and Professor Shanmuga, whom you've heard earlier today morning, right? And the whole idea is you want to reconstruct images that the person is seeing using their brain signals. Now, obviously, we are not the first to do that. You can use this encoder and classifier approach. You can have different types of deep learning models, CNNs, LSTMs, encoder, decoder architectures, right? Generative discriminator. I'll not go into all of those details, right? So what we have proposed, okay, we use two architectures, LSTM and CNN, nothing fancy there. What we are trying to do is, if I give you EEG signal, okay, can you sort of learn the representation of this EEG signal? Because that is what allows us to generalize. Now, obviously, when you have LSTM, it's more suitable for time series data because EEG is a time series. You might want to prefer LSTM. But nevertheless, we did have decent results from both the architectures. So this is a data set from 2017 CVPR. It has 40 categories of images. And when we train EEG data using triplet loss, so the triplet loss is like, so the triplet loss is like you have main data point, you have a positive sample, and you also have a negative sample. So what you try to do is that's why it's a triplet. So you try to bring the positive sample and the data point closer, and you try to move away the negative sample and the data point. So you are trying to ensure that the similar data points are all together. And that, when you look at the embeddings, you clearly get the separation. That means the representation is learned, that these classes are all now separable. Then you can use it for classification, you can use it for GAN, whatever function you want. So we also had some issues where it always does not work. Like in another data set, it didn't work. And in third data set, the LSTM worked, but not the CNN. So we are able to basically get the SOTA with 98% accuracy in the 40 class identification. But obviously, it does not generalize to all the data sets, although you train it from scratch. And one of the data sets where we will not able to do is the object data set. And on the thought which is basically you are visualizing the objects, there we get not the SOTA, but a decent accuracy. Now, what is the interesting part here? Okay, whether you say I do classification, you do generative adversarial networks, you do image generation, these things are okay because you're training on a data set, you are reproducing on it, or maybe you're testing on multiple data sets. The interest in learning comes when we are able to do what is known as zero-shot classification. That means something that you have never ever seen, object categories in your training data. So if you did out of the 40 classes, we took away 6 classes. So only 34 are used in training testing, 6 zero shot, right? Only testing. So it's not like, so you have not seen even a single instance of those 6 classes, right? So you have seen bottles, pen drive, books, pens, and so on, but you have not never seen, let's say, cat and a bird, and so on, right? So we are trying to see if those generalizations do occur, and that's what you see. So as long as the representations are separable, they form different clusters, we can say that we are successful in zero-shot classification. And again, we have very good zero-shot results. So the image generation part, now that the representations are learned, right? You again can see the different types of images that can be generated. You see the top 5 images generated, and they are fairly representative. There is this inception score, which is the standard now for GANs. We use that, and again, ours is the best model. Now, another way one can think about image to image translation is using what is known as the clip architecture. So here what you are trying to do is you have EEG features, you have the image features. You try to maximize the diagonal and you try to minimize the off diagonal. So the alignment between the two modalities is what is being maximized. And the hope is that, again, for zero shot approaches, generalization will be very good. So that, again, here you have the ground truth and top 5 retrieved images. You can see how well the joint representations are learned. And that's all I have. And yeah, Prajwal is there at back, who started all this work. And that's the team. And I'm only doing the presentation for you. Thank you. Questions? Yeah, yeah.
So I was wondering about the neurovisual signatures we have for different diagnostic groups of people, like unhealthy patients who have dementia or Alzheimer's, and healthy group of people with no such problems. So when they are looking at an image, what kind of, or even if they're not looking, what kind of an neurovisual pattern will be formed in the brain? Is there any research done on it, and what do we know about it so far?
So one of the problems with the brain, or one of the advantages, if you want to see the way it works, is advantages from the perspective of how robust our brains are. Like you think about what happens to the visual cortex of a blind person. That visual region is repurposed for language, music, touch. So what is happening is there's a great amount of plasticity, right? And that plasticity is enabling us to recover the function. And that, from a biological perspective, is an advantage. From a machine learning perspective, it says that every person's brain is different. It is shaped by our experience. So, although the structure of the brain, right? We all have the same lobes and sulci and gyri, and they are all. It's like your hand, right? The function is the same, but they are all distinct. So that poses a problem if you want to build machine learning algorithms that can generalize. Now, common to a particular disease like Alzheimer, we know, let's say there is a memory loss. So MRI scans can give us the deep brain structures that encode this. But also in the EEG patterns, or in the brain connectivity, connection between the different regions, there could be slight changes that again can be used by your machine learning algorithms to be able to classify the specific type of brain disease one person has. And this you cannot do by direct medical diagnosis. You can do with what is known as neuropsychological testing. That means you observe deficient patterns in the brain, like somebody can speak but not hear. Okay, that is a behavioral pattern, and it's difficult to localize their deficiency. But if you are looking at the brain activity, then it's possible to build automatic algorithms can classify the disease.
Okay, sir. Thank you.
Understood everything or nothing? Only two possibilities, no questions. Or it's too much to take. Anybody working on brain data? Emotion identification? No? You worked on?
I'm working on a similar line. The objective is to try and understand the affective and cognitive relationship while playing a video game on the children. So whenever we do some cognitive task, there is the foundation is that there will be emotional intelligence. So once you have the emotional intelligence, can try to have a relation with what happens to the cognitive ability. It's getting higher, listen, positively publications. So that's how I'm trying to answer my research questions in this domain.
Yeah, nice, interesting.
Thanks.
You can see, when you get more points, the person is happier,
Right.
right? And when you lose a life or something like that in the game.
Yeah, the element in the game, like when my character will die, I will get the negative emotions.
Yes.
at that time I may not be able to handle the different elements in my game. So that time the cognitive demand will be so high for me.
Yeah.
I mean, that's the angle I'm trying to look.
How many channels you are using?
Currently, we are having 32 channels CEG,
Okay.
products.
You can also try with single channel.
Yeah, that will be looking into part of the.
So, especially with applications like gaming, you don't do it in the lab, right?
Right.
So, you want to have something that people can use in their homes and so on.
Right. Sir.
So, one possibility is after you do the in-lab testing, you extend it only to maybe a single channel device.
Right. Like as per the literature, I'm trying to find the relationship, but then I want to validate on my subjects and all. Yeah.
Okay, we'll stop here. All right, thank you.